do a quick introduction to Dr. Daniela. She's a psychiatrist and also is a psychotherapist, and she works in the child neuropsychiatric unit at St. Andrea University. Most recently, she completed a PhD focused on sensory neuroplasticity, specifically examining the impact of early adverse experience on neuropsychological development in infancy, which is pretty close to our work. And her research was conducted at the unit of pediatrics. Within the Department of Neuroscience, Mental Health, and Sensory Organs. So Dr. Polese is currently leading a project on neonatal brain plasticity. So today she'll share her work on human birth theory and neurobiological correlates in newborns' reaction to light at birth. And it's our great honor to warm welcome Dr. Daniela Polese all the way from Italy.
Thank you very much, Guy. It's a pleasure and an honor to be here with you. I'm trying to show you how we are building our research on human birth since the beginning. So let's start from, I don't know if there is a microphone for the online.
I think it's actually.
Perfect. Yeah. Okay, great. So I would like to start from this video, and I invite you to watch it a little bit in detail, focusing on it, and we will get back later on this, okay? Okay, so I can introduce you briefly to beginning the human birth theory. It has been formulated in 1971 by Massimo Fagioli, an Italian medical doctor, psychiatrist, and psychotherapist as well. And he considered that there is a crucial passage from the time before birth, the period when the fetus is in an homeostatic condition with darkness, silence, immersed in the warm amniotic fluid, and the afterbirth, when he considers a light stimulus that catches the brain and determines a brain reaction during that 20 seconds of silence that we have just seen in the video. It was a little longer before. And he also stated about a simultaneous mind and brain activity. So we consider that there is a capability to imagine at birth, and then there is the first breath and wail. So it's interesting also for psychosomatic disorders to see that he considers body and mind and brain going together since the beginning. But let's see now, let's go backwards and see how he thought about this theory, because it's very, very important. It seems to be far, but we will see how it goes. So how was the human birth theory developed? Massimo Fagioli, that you see in the picture, trained at the neuropsychiatry unit at University of Modena, and he started working at psychiatric hospital in Venice. So imagine a psychiatric hospital in this island in front of Venice, Piazza San Marco, and he was there. It's about 1959 at that time in Venice. Then he worked at psychiatric hospital in Padua, where he started bringing the patients out, because there was this very particular period around the world where patients were starting breaking out because psychiatric hospitals were transforming into psychiatric communities. You got a big one here in Maryland, the Chestnut Lodge, Rockville, where Huck Sullivan and Frida from Rackman were working there. I don't know if you're familiar with this, but they were very, very important psychosis psychotherapists at that time. So he worked at Kreuzlingen Sanatorium in Switzerland, which was a community at that time. So working with severe mental illnesses, such as psychosis and schizophrenia, Fagioli particularly focused on the delusional perception. Delusional perception was firstly described by Karl Jaspers in 1913 and better developed by Kurt Schneider in 1946. What is delusional perception? Is a core symptom previous to delusions. And it is the abnormal attribution of a meaning to a common daily stimulus, a perception. Let's go to a very quick example. There is a red car pass by, and delusional meaning is attributed like, that red car means the CIA is watching me. Okay. In one of his first works, Fagioli describes particularly the immediacy of the delusional attribution of meaning. So he highlights that the delusional attribution of meaning is simultaneous to the act of perceiving, simultaneous to the stimulus. Like this. Red car means the CIA is watching me. So reaction represents the core process underlying the thought activity, and it includes the simultaneity. So he just noticed this reaction, which, you know, you can say, oh, that's a reaction, but he really focused on this process of reaction about thinking. And then there was another novelty that he proposed, the simultaneity of both perception and meaning attribution as a reaction to an environmental stimulus, as I said, but also differently from classical psychopathology. In this reaction, the sensory is not altered. Because still now we think that the hallucinations are the alteration of brain alteration, and also delusions and delusional perception. But he thought that it was not an alteration of sensory, but a thought alteration. That is a mind alteration. So thinking that that car means that the CIA is watching me. During psychotherapy with psychotic patients, Fagioli, this is a second observation after the reaction and the delusional perception, he observed specific reaction to the physical absence of the psychotherapist, generally after holidays, specifically after the summer holidays. And the patient came back as if the therapy did not exist before, with the same symptomatology. Hopelessness, severe sickness. So the patient led the therapist believing that that improvement never happened. But he formulated this conception of annulment pulsion directed against the therapist, and it resembled a psychological blackout of the relationship with the therapist and the therapeutic progresses. So first, delusional perception as a pathological reaction to a common stimulus. Second, annulment pulsion as a pathological reaction to the absence of the therapist. What about the physiology? What about at the beginning of our time when we are babies? And he started working at the unit of child neuropsychiatry at Sapienza University of Rome with Professor Giovanni Bollea. And he worked with autistic babies and also with infants, looking for this original mind functioning. So how does the mind work from the beginning? Physiology as a natural functioning, physiopathology as a loss of natural functioning, and then the therapy is seen as an intervention in order to restore the natural functioning, which was at the origin when we are a baby. Then he formulated the human birth theory in this way. The title of the book is Death, Instinct, and Knowledge, because at that time it was totally in contrast with this idea that we have the death instinct when we are born, because psychoanalysis was used a lot in the child neuropsychiatry unit. So it was totally in opposition to this conception of original perversion and all this stuff, okay? And then he wrote, by observing reality, we can reason that the infant, when he or she is born, comes from the darkness and the water of the intrauterine condition. The newborn comes, and it sounds logical, from a previous stage. We can therefore assume that the newborn will react to being expelled to bombarding stimuli in the new situation that shock him or her by wishing and or fantasizing about returning to the previous stage. Then he writes, the truly new condition, which previously was absolutely and totally new, non-existent before, it's only light. All other senses, for example, taste, the sense of smell, touch, and synesthesia, they have all been stimulated in the uterus, with the exception of hearing, as the fetus may not hear in the uterus, may not hear sounds properly, because there is the amniotic fluid. So at birth, the only part of brain which is directly exposed to stimuli is the retina. We can say noise, light, and cold. But given the intrauterine dark, light is absolutely new stimulus, which reaches through visual pathway, the occipital cortex. Brain reacts to light through a simultaneous activation of brain areas. So you can get the connection between the concept of reaction, simultaneous reaction, that we got in delusional perception and anomalous passion before. Brain starts a new functioning and activates organs and apparatus for living. So we can go back to the first slide, and we can see how there is this before birth and after birth, and the light stimulus and brain reaction during 20 seconds of silence, and for him also a mind functioning, featured by a capability to imagine. So Fagioli acknowledges qualities for babies at birth, such as capability to react, vitality, affectivity, capability to imagine, and interhuman relationship. Let's consider also that vitality was already a biological concept, and it's still measured by the Apgar score by the anesthesiologist Virginia Apgar. And this is just a spot for you. This is the National Gallery of Modern Art in Rome. And we got this written on the stairs in 2020, 2022, which was also nice because it was a kind of hope during the pandemic. And it means the roots must trust the flowers, written by the philosopher Maria Zambrano, she's Spanish, and it's about the babies, that we need to trust the babies. They can be more than us, actually. So the book, Fagioli's first book, was translated in several languages: Spanish, German, and also French, Portuguese, and English. Now, let's go to the second part of our speech today, neurobiological correlates. About the seconds of silence, World Health Organization guidelines say that first breath can physiologically occur up to 60 seconds after birth. This delay varies interindividually, obviously, but averaging to about 20 seconds, and has been well documented by several reports. This is an interesting art study by the photographer Christian Berthelot, which is made specifically on Caesars, which is very, very Beautiful. So we proposed to the scientific community a debate with this paper. Something must happen before first breath. So what's really happened during that seconds of silence before first breath? It's not generally studied, so it's pretty new. We know that there is the first respiratory action that must necessarily be presented by the clearance of the fluid from the lungs. So there is, during labor, already an adrenaline mediated activation of sodium channels. Then the secretion of the fluid is stopped on the apical pulmonary surface. But then we need the diaphragm contraction to complete the clearance of fluid, okay? And then the first respiratory action. So we asked, as in every respiratory action, the diaphragm contracts and flattens so that the chest cavity enlarges. This contraction creates a vacuum which pulls air into the lungs. And this contraction needs an input. It cannot go, alone and it's the biggest respiratory action of our life. That's very impressive, right? So how does that diaphragm contraction happen at birth? We have checked the literature and about the sudden death in the crib, we have found that an abrupt activation of the Colliger-Fuse-Pontian nucleus at human birth happens. That means that at the sudden death, this activation is lost. So you have like an inhibition by the brain to the respiratory activity. And we found that there should be a primacy of central nervous system before the diaphragm contraction. So what does happen before first breath? There is a CNS activation, a brain activation, and then the first breath and wail. Just take a little bit of water. Which is the candidate that can trigger this brain or CNS activation? We have studied a lot of literature, tons of literature, I have to say. And our hypothesis was that the Newbert's reaction to light is determinant for the brain's activation at human birth. So we proposed this hypothesis and theory article with the University of Siena, with Dr. Maria Letizia Riccio, with Massimo Fagioli as well as last author, and also University of Tor Vergata, Alessandro Mazzetta, and Marcella Fagioli and Francesca Fagioli from the National Health System as Rome 1. So given the changes occurring from the prenatal brain to the postnatal brain, we aimed at characterizing the very early stimuli that triggers these changes. Among several diversified candidates, one single specific activating agent only is characterized by the above mentioned qualities, which are novelty, because it's very, very new in the extrauterine environment, impact. It's very strong because it immediately gets the central nervous system because it's energy, it's not material. So you have stimulus and reaction very, very quickly. And ubiquity, because if it's connected to life, it should be everywhere, even in the dark, because in the dark there are photons, the absence of photons do not exist in life. Moreover, we can speculate that this stimulus can reach the brain directly without any intermediate transfer of information, that is light. Then we got this article about period of gestation when the visual connections are complete, almost complete. are formed, I would say, and it's about the 20-22 gestation weeks. Okay. This period, 20-22 gestation weeks, is the period when there is the viability for the fetus. That is, the fetus can survive out of the womb. So it's exactly the same period. There is an overlap. And at the same period, the subplate is completely formed. There is a functional maturation of the subplate at this stage, as Professor Gatti with her group and Fagioli too wrote in 2012. And so the subplate was discovered by Kostovich before, and this connection let us think that the visual pathway is connected to the survival outside of the womb. So the possibility to live is connected to this pathway, which can be connected with the first breath. So it matches. Okay. Then another article by Van der Waal and Montreal was very, very astonishing. They say that they were astonished when they realized that the 3 blind people were able to perceive the light without being aware of that. So here came up that there was a different pathway connected to the optic one, but it was not really visual. And it's been called the non-image forming pathway, NIF, or non-visual pathway. And it was connected to a new opsin, not rods and cones, but a new opsin called melanopsin, which was discovered before. And it's characterized, it's present in the IPRGGs, which are the intrinsically photosensitive retinal ganglion cells. Okay, so that's a very beautiful title here. Retinal development, second sight comes first, because this pathway was more developed at birth than the visual one. You know, the newborns don't see clearly. They see, but they are blind over 20 centimeters. I don't know the inches, but it's very, very close. Just the distance between them and the mother face, then they don't see anything. And even the face is very not clear, not clear. So let's see, the retina is the unique photoreceptor structure of our body, and it's characterized by several layers. And until a few years ago, an absolute belief dominated among researchers that the retina had only 2 types of photoreceptors, cones and rods. But IPRGGs are a very small fraction of RGs, around 1%, and unexpectedly, they are not an homogeneous population of cells, but they are include several subtypes. They are very sophisticated, and they are active at birth. So in our work, we highlighted that there is, you can see the dark gray, the non-visual pathway, while the clear gray is a visual pathway, and it's active and mature at birth. And this pathway directly projects to several brain areas. It's very, very widespread. It's impressive. Consider that IPRGGs have long axons. So they directly project from retina to the thalamus. So they're the core of the retino-hypothalamic tract. So they can really activate the whole brain through the thalamus and the hypothalamus. And they also project to locus coeruleus, and they can give this immediate brain activation. This is very interesting because the amygdala, you know, obviously, amygdala is a sublimbic area and is involved in emotional processes, also receives direct projection from the IPRGGs and might represent the key target of the non-visual pathway by potentiating effects of light on alertness and mood. These limbic areas is part of the neural network named the salience network associated with responsiveness to stimuli. There is this contemporary concept of delusional perception that is called aberrant salience, which is the abnormous attribution of meaning to a neutral daily stimulus. And it's located in the same network. So it's impressing how this process, which is the delusional perception, which triggered in Fagioli the research on birth, now they matches neurobiologically. That's very, very impressive. So there is a normal salience connected to the reaction to light at birth, and also an aberrant salience and pathology. But for Fagioli, the primary abnormalities is in mind and not in brain. That's very important. So brain is a correlate, because we are also body. We are made of mind and brain together. So he distinguished neurology from psychiatry in this sense of cause psychopathological and pathogenetic causes. So we can see how these seconds of silence have been studied. Here, this group, this is an Italian group, and they studied these seconds to get the first breath after birth and after labor. And it's interesting also, this other study by Polli in Milan, published on Nature in 2010, where you can see that photons can immediately, simultaneously activate the retina in 220 seconds. And femtoseconds. A femtosecond is 10 minus 15 of a second. So it's a very, very short time. We can say there is no time. So this is a confirmation of this, a strong correlation with this immediate reaction, retinal reaction to light at birth, and brain activation with the first breath in a few seconds by a brain input to the diaphragm. And there is another thing very interesting, and I know that you know this because Moriah talked about this when she came in November in Sapienza for Health Talk. There is a GABA shift that no many researchers know actually, but you know, because in the fetal period, GABA has an excitatory functioning, which is very impressive since the GABA is the main neurotransmitter for epilepsy treatment. And we use obviously a medication which potentiates GABA functioning to inhibit the epileptic seizures. But during the fetal period, it's excitatory. And when this change of functioning happens at birth, that is another factor that led us to think that at birth there is a strong revolution neurobiologically. So in our paper, we summarized the findings in these slides. There is a dark part with a period before 24 gestational weeks when the possibility to survive and live occurs, which is viability. And when there are the formation of IPRGC's tract, the non-visual pathway, and also the GABA shift, the shift and the subplate formation, and also the retinal hypothalamic tract formation. This period is characterized more by spontaneous neuronal activity, even if, as I will show you after the 24 gestation, there is a sensory expectation by the brain and not before. Before we are like totally closed in a box. But in this period, the stimuli are mediated by the amniotic fluid. So all the sensory driving is due to the endogenous stimuli. While at birth, we have this sensory dependent activity totally, because there is a strong input from the external environment, and also we are not in the amniotic fluid anymore. And we are independent. We are an independent organism by the mother. So we directly are in relationship with the nature, light, cold, etc., and then also with humans. Then from birth, there is the IPRGC is functioning, and this CNS reaction to light via IPRGCs. Then GABA is, it's not really a direct shift. There's an oscillatory shift, which suggests like a modulation of the response to light at birth, of the response to the new environment at birth. It's a modulation that's very intriguing, right? Here's a very, very recent work by de Groot. It's in Utrecht, in Holland. And he said it is about sleep, but it basically highlights that there is a sensory independency before 24 weeks of gestation. And then we start to be sensory expectant. But the sensory stimulation, you know, is dependent by where we are. And also by the brain condition, because when the brain is still in the fetus and the fetus is still connected to the mother, the brain has not the same functioning. Indeed, we have, for example, the Colliger-Fuse-Pontine nucleus not active. So it's very different functioning. It's really spontaneous functioning. Then there are a few, I think you should consider these papers in your work, because there are only two studies, according to my knowledge, that directly they talk about the impact of an extrauterine environment. And for example, in this study, they compare the preterm newborns, brain functional connectivity to the fetus of the same gestational age, functional connectivity, brain functional connectivity. And as you see in the red lines of many brain areas, the brain functional connectivity is higher and higher in the preterm newborns. That it should be due to the extrauterine exposure, which is a very big stimulation. The second paper compared the very preterm babies to late preterm babies, and this shown that the very preterm baby take advantage from the exposition to the external environment, which also connected to your paper about the early preterm babies, because there is some brain changing during birth, across the birth in a different way.
Yeah.
I was very impressed by that result, as if that brain is looking for the external environment as something is not going on in the womb. So the programming is changing to look for this external stimulation, which can help at a certain stage, obviously not too preterm, but it can help the brain development. And this study shows how there is an acceleration of the brain development thanks to the contact with the external environment, where there is a reaction. So the concept of reaction is crucial. So you can see here, in this paper, we made a big work on a private birth video with consensus, etc., because we captured the frames, several frames of the birth video, and we chose these two ones because the first one is 4 seconds after birth on the left, the A. The B on the right is 12 seconds after birth. You can see in the first one, we catch the moment when there is a cavity on the chest, which is due to the collapsed lungs because the air is not yet inside. The skin is pale, the baby is cyanotic, and you see the muscles are hanging because there is no tone yet, because there is no breathing. So it's supposed to be the seconds when there is a light stimulation and brain reaction and activation, but not yet the brain input to the muscles, to the respiratory muscles. Then in the 12 seconds one, there is no change, you know, and there is no filter. This is like you see the both pictures. So we didn't exaggerate the effect. It's really how it is. And in the second one, the pink is, the skin is pink and the muscles tone is physiological, is normal, is healthy. And the chest is, there's no cavity in the chest anymore because the lungs are inflated. And we are going to see the whale. Now there is already the first breath. And Fagioli characterized this phase with these words: reaction, pulsion is not the annulment pulsion, is a vital pulsion because it's with vitality. So reaction, pulsion, vitality, creation, existence, time, capability to image, and strength. It's a bit complicated, but I want to mention those words to you just to know. And then there is the whale. This is about 18 seconds. Okay, we have the sound of the whale, the movement, etc. Okay, so this is to summarize. We have also some species-specific features. For example, the unique pattern of rotational girth, and also an elevated head to cervix presentation during labor, during labor, and also this impossibility to move, because, for example, primates and other animals can walk, can actively from a motoric point of view, can reach the breast, while we cannot. So there could be a mind compensation activity as human beings. Okay, so birth now is a very powerful moment. It's a powerful step. And we are working about this with different groups. And I had to mention your work, that I think it's amazing, really amazing. When I saw it, I was very impressed that you published it. And it's very interesting that you wrote this, that you talk about a surge of functional connectivity, probably likely due to the external world. It is very correct. It's very, very correct, in my opinion, obviously.
I agree.
Yeah, absolutely. So you know this study. And I wanted to open brackets about our sleep lab. Just a couple of slides. Because Professor Oliviero Bruni founded the sleep lab in University Hospital Sant'Andrea, and I work there. I work a lot on sleep disorders on infants, the first year of life too, because we identified that there is also a disorder during the first months of life, and the pediatric insomnia has a 20-hour impact with a negative consequence on daytime functioning, both on baby and parents. So it's a very, very delicate topic, and is very frequently overlooked by clinicians who do not diagnose pediatric insomnia. And 6 months is a reasonable age to consider a diagnosis of chronic insomnia disorder. You can see the different types of insomnia: difficulty initiating or maintaining sleep, early morning awakening, several awakenings during night. And we have Professor Bruni identified 3 different types of insomnia. For example, one is connected to iron. I know that here you study iron, brain iron a lot. See this movement?
Run. Go Wyatt. Go Jasper. Bust a move. Bust a move.
So it's lovely, but this
Bust a
movement is
move.
a restless leg syndrome, like syndrome. It's impressive how with iron,
Run.
this movement
Run.
disappears. And night changes totally.
Go.
Pilots come back like saying,
Go,
bless
go,
you,
go.
bless you, because they can sleep.
Go.
Simply with the iron at the correct dosage, obviously. Like, it's not even a medication.
Babies.
So, firstly, according to what we have mentioned before, the sleep lab brackets, considering birth as a revolutionary step, we are writing with Moriah and also Dr. Bibi at Columbia University and Dr. Doug Alice in Harvard University, a perspective paper about an alternative hypothesis, because until the last decade, there was a conception that considered birth as a starting point, in the sense that we were like tabula rasa at birth. We start our experience at birth, and only at 4 or 5 months, you can check our skills, because it's like we are nothing at birth. How can I say? We start with a blind situation, like lumps of clay, I would say, as a traditional vision of the newborn at birth. So the BICE, which is a group from Colombia, they proposed perinatal continuity. So not birth as a starting point, but the last trimester as a starting point, with a sort of continuity and just a recalibration at birth. Okay, so we are writing a paper with a third way of seeing the very early development and considering the birth of a crucial point, like a cutting edge. Not because before there was nothing, obviously, because we have seen there are many necessary processes before birth, otherwise there's no birth, but at birth, the impact with the extrauterine environment breaks everything was before, including, for example, the GABA functioning changed totally. And we would like to propose this at the scientific community together, the four of us. Then there is another work in progress, studying newborns' behavior and sleep. And it is with Columbia and Harvard as well, and the Hospital San Camillo in Rome. We would like to investigate in a population and sample of newborns, the physiological change occurring during the time interval before the first breath. So the behavioral features at birth of the newborn, before first breath and right after it, and also the sleep characteristics right after birth. Because since Professor Bruni is a world expert on birth, we couldn't lose this opportunity to study also the very early sleep. So now we can see the same video according to what I have just said together, okay?
Fate sentire mamma. Ah, che bella donna. No.
He's saying, come on, breathe. Why don't you breathe? Relief. Okay, and you can see in the other picture the same baby sleeping right after birth. Okay, and this is Moriah and Casey. I think you have met Casey, and I met them in 2022 in New York. Then Moriah at Sapienza in November. And the center, there is Professor Oliviero Bruni and Francesca and Michelangelo, neuropsychiatry residents. And on the right, there is Professor Pasquale Parisi. I'm working with the newborn's reaction to light. With Moravia, and also Professor Alessandro Ferretti, and my great collaborators, which are Mattia Costa and Alice Rubeo, who I thank you so much. Thank you to you for your effort. Okay, was it? Okay, good time. Perfectly. Okay, please.
Question,
Absolutely, questions.
Mark, nice to meet you. It was really, really interesting. I just had a question about the functional connectivity in the premature birth, that paper you were talking to Lanxin about, the in utero versus ex utero premature birth functional connectivity. When I was wondering when it was taken, the functional connectivity, like post birth, when was it, like at what point? 10 points.
You mean the studio about the comparison between the preterm with the...
Yeah. I'm just wondering when the MRI would have been taken for the functional connectivity.
Ah, yeah, sure.
At what time point?
Yeah, I think it's...
Like how far post?
No, it's all the same age. I think it's it's like 35 weeks. I think it's 35 weeks.
Yeah, so that might be a couple of weeks.
Yeah.
Post birth for the.
Yeah, exactly. I think, yes. I should control, but I think 35. Because 25 is too early.
Yeah, I can look up the paper.
Yeah, but I think it's a very good point, because they wanted to be sure that the age was the same for all babies.
Yeah.
So they catch them at the same point. It was the preterm phase, I think it's 35, because if I'm not wrong, they wanted to compare the very last period of the last trimester and the birth, after birth.
Yeah, that's
which is more difficult to find the differences, right?
Yeah.
Because I expect more differences between, for example, 25 weeks and after birth.
Yeah.
But the 35 is incredible that there is differences. No?
Yeah, and that's why I was asking as well, because your whole post birth, that like the week by week basis, nearly like with the brain development. So I was just curious as to when it was taken.
Yeah, check the paper though, because you can be sure, but it's the same age. It's a late, not really a very preterm, like the other paper, which compared the late preterm with the early preterms. Thank you very much for your question. Please.
Thank you so much for the talk. This was really, really cool. I guess I had a question about the IP or GCs. Are there any groups doing animal research on that pathway? And I'm curious if it's evolutionarily conserved. It seems like a really interesting, like, biological mechanism. And it's also really cool that it's somehow pre-conscious, like there's no conscious perception of light.
Yeah.
I wonder if any groups do work in the animal research and if they're knocking out or manipulating that circuit at all.
Yeah, there are many research groups working on this in animal models with this pathway. If you read our paper, we mentioned at least until 2022, the middle of 2022, we collected a lot of research in animal models. The point is that we should consider also in the reaction who reacts. Because I expect that there is the same or very similar reaction at birth in animals, but animals' brain is not human brain. So humans have this capability to create, to invent things, to discover. We are animals, but we are not really animals, because there is a gap between us and animals. So we have to consider that there should be a specific human brain response to light at birth. So you can study the IPRGGs, but you need to study the human brain. That's why your group is so cool, because you are, I think, the people most in advance studying a human fMRI in fetus and newborn, not only fetus. And that's why I was like stalking Moriah a few years ago, because I study groups who were doing this, and I found that she was almost the only one really doing this, because she suggested me one group in Italy, but it's not the same. We are really focused on the human brain functioning and to the development between fetal stage to life. Okay, so newborns. So you can study that those researchers, but the human studies are even more interesting because you have to always consider the two factors of the relationships. At birth, you have, before the human brain, a relationship with the nature, which is not human, not animate, like light. Then there is the human interaction. So human being, human brain, a newborn's human brain, with caregivers, mother and father's brain and person, and you have the human relationship at that point. So it seems simple, but it's crucial because sometimes we are lost in many data. So you lose the point, we lose the point sometimes. Okay, you're welcome. Thanks for your question.
Thank you for this amazing talk. I'd love to hear more about what you think around the sensory perception before birth. So maybe a long time ago, well, not that long ago, but we used to think that the fetus is learning before they're born, and then it's epigenetics and genetics that take over in how we think, we become who we are before we're born. So the idea that you could learn anything before birth is kind of forgotten about. And then we have this DOHAD prenatal programming idea that still mostly focuses on epigenetic mechanisms and biology. But there is a kind of a small but very interesting field that has continued to try to understand sensory perception before birth and whether there could really be learning. It's tricky to study, but I know there are a few that look at sound and even light and response to taste in utero. There's some really cool, interesting methods to study these things, but I'm just curious what you think.
Yeah, I know obviously that studies, but the point is that we are a little bit dependent on our beliefs.
Yeah,
that's the point, because we like to think about talking to the baby when he or she is in the womb. Music. It's very romantic. It's very nice.
Hear mom's voice.
Yeah, it's written everywhere so far. It's still believed, but we need to be more like you say, lycistic, like without belief, not ideologic.
Agnostic.
Yes, atheistic in some way. I mean, scientifically, okay, because the studies which would like to demonstrate this, firstly, are studies that are based on the idea that the stimulus, auditory stimulus or the visual stimulus, can get the fetus. And they take the confirmation that this stimuli get the fetus because the fetus moves. But the sample population is very poor. It's like 3, 4, 5 fetuses. And the movement is not enough to demonstrate that that's something, because there is a spontaneous movement. And also you have a movement which is induced by the mechanical synesthetic stimulation. I will give you an example. There is a study with the Ninth Symphony, Beethoven Symphony, the hymn to joy, which is a strong sound. They put in the vagina to be sure that the fetus can get the sound. But this sound is transformed into wave into amniotic fluids. So the main stimulation which gets the fetus is on the skin. Unless you don't do an fMRI and you don't detect an auditory brain activation, you cannot be never sure about it. You would love to think about it, but you cannot know that. And the fetus reaction was very strong. That fetus has spontaneous movement, also the mimic, the facial expressions. So they recorded the facial reaction, which is still mechanical. And they said, he's trying to talk. He's trying to answer to the sound, but it's totally an interpretation.
Unless you have the same sound very soon after birth, there is like 1 or 2 studies that think they can maybe remember the sound.
It can be.
Yeah.
Can be.
It's very hard.
No, I think that it can be according to the skin stimulation. Because when we are born, we have a skin trace on of the contact of the amniotic fluid. There was a sensory stimulation during the whole pregnancy, and especially when we had the sensory activity possible in the last trimester. We really have the trace of this. We have recorded this trace, but it's more peripheral. And then when the brain is active, can catch this peripheral stimulation. Because until you do not detect a brain activity which is not spontaneous, it's always an interpretation. So these are kind of recognizing the music, if it is, because there's this correlation is a bit hard to demonstrate scientifically, really, but it can be a mechanical stimulation by the sound. Beethoven was, he couldn't hear. He became, I remember now, the deaf. Thank you very much, deaf. But he distinguished the music with the mechanic sound waves on the body. Yeah, it was Beethoven, obviously. We cannot do that. We can't do that. But just to say that we need to be very careful. Very often we project our expectation and our thoughts on the baby, and the womb. I know it's very common, and this belief, it's very common, but it's not really demonstrated scientifically. Also with the baby, when the baby is born, we interpret a lot his movement as if he or she was us. That is a newborn, and then is a baby, an infant. So
if I can add too, I
Please.
wonder with the music exposure,
Please.
if it's even, mom listening to something she loves and having that moment of joy is always going to help the prenatal environment. Same as talking to her baby, and so
totally agree with you.
Yes.
But this is not seen, because I want to
Exactly.
see that he's responding to the same music. While there's an environment, emotions, so he's happy because the mother is happy. That's a very, very brilliant point.
We do know that doesn't matter, even without sensory stimuli, that in utero babies will react to mom's stress through some other signal, like respiration, heart rate, and that it's not, the baby may or may not be experiencing anything except the mom is feeling differently, and you can use ECG to see that over time. In Catherine Monk's lab, that's what we studied, and it's
Brilliant.
some other signal that's not
Brilliant.
sensory.
And I think we have also to remember that placenta has a protective system based by proteins, which defend the fetus from the higher level of cortisol and the mother's blood. So there is, a certain point until a certain point, the fetus does not have the consequences of the mother's stress. So
Unless mom is depressed or anxious.
too much. I mean, you have to see, because there are many factors in the mother's organism which are connected to the depression or anxiety, but there is also a strong defense of the baby. Otherwise, we would have never developed so much, because I can say that pregnant mother is always stressed. Really, it's impossible that during your pregnancy you have no stress. But there is a pathological level that probably changes things when you should detect it, and the blood values as well. And I can also show you this other, that's the point, because I was leaving this these slides. There's starting a relationship here, which is not voluntary, aware, and necessarily conscious. And we published before this review with a group in Lille, which is connected to Sapienza, with Professor Maccari. And they study the effect of stress, the epigenetic effect of stress. Okay. And they demonstrated that there is a difference even in animal models, if the offspring is adopted. They exchanged the offsprings between mothers. So they gave the puppies of a stressed mother to a not stressed mother, and the opposite. And they got that the stressed puppies were the ones who were nurtured by the stressed mother. So this is another point. If I study an infant, I cannot connect to the prenatal stress, mother stress, because I cannot be sure that there are other steps after birth, so the contact with the stressed mother, which are the real risk factors for stressing baby. Because again, there's like a belief that there's something before, so I'm blind about the step in the middle. So there's this connection between the prenatal and the baby at like 1 month, without considering all the days spent with the mother, which is very easy to understand how he got this influence by the mother's depression or mother's anxiety. And it's very important that we try to change this conception of the 1,000 days of life, because it's considered from the conceptional age, as if the baby starts there. While at birth, obviously the pregnancy is very important, it's pivotal that you care about pregnancy for the future baby, but there is something that begins at birth. So when you're talking about babies, we should be very careful to talk about something that can happen from birth on. Okay, I think, yes, that's very interesting, because the referee here asked about the human birth theory. So they developed the paper into 2 parts. And the second one is about the human birth theory and the development. And they wrote that they think that epigenetic consequence of prenatal mother stress can be reversed from birth with the adoption. In my opinion, it can even be that the main actual influence can be seen from birth. So we have to be careful about the prenatal epigenetic alteration. We have to be free from our beliefs, I think. Okay. Okay, no more questions? Okay, thanks for your work. See you.